Okay, so these are the problems that we did in class, or that you did in class, from that handout. So when we look at free earlobes are dominant over attached earlobes. So I'm going to use F representing free earlobes are dominant, and then recessive are attached. Okay, what will the F1 offspring be from a cross between two heterozygous free earlobed parents? Okay, so the parents are heterozygous. And what we have here are the parental generation. Now our F1, when we look at this, here we go. Okay, so our F1 offspring, we have a homozygous dominant, we have two heterozygous, and we have one homozygous recessive. And so when we look at this information, we can do two things with this. We can find the phenotypic ratio, and in the phenotypic ratio, there's 3 individuals that have the dominant phenotype, and 1 individual that has the recessive phenotype. So if this was asking for your phenotypic or genotypic ratio, this would be your phenotypic. Now for our genotypic ratios, what we see, if we're looking at genotypes, you start with whatever is like homozygous dominant, then you go to heterozygous, and then homozygous recessive. So the answer for our genotypic ratio would be a 1 to 2 to 1. Okay, the ability to taste PTC is dependent on a dominant gene. So we're going to say that tasters are dominant, and the recessive allele is associated with non-tasting. Okay. What are the possible genotypes for tasters and non-tasters? Well, tasters can be homozygous dominant or heterozygous, and then non-tasters can be homozygous recessive. Two parents, part B, two parents, both tasters, have, so here's one parent, there's the other parent, they're both tasters, so we know they have at least one dominant allele, okay, and they have an offspring, so this is the parental generation. Now in the F1, they have a homozygous recessive child, a non-taster. Okay, so how do two parents that are tasters make a non-taster? Well, they would have to be heterozygous because they'd each have to give one of these alleles to their offspring. Okay, so the genotypes of the two parents are both heterozygous. Now for part C, two parents, one taster, okay, and the other a non-taster, have a taster offspring. Cool. What is a genotype of the offspring? Well, for this one, we know that they're a taster, so they'd have to have at least one dominant allele, and that dominant allele they got from this parent. Well, then the second allele they have to get from their other parent. Well, this other parent only has recessive T's to give, so the answer for this one would be a heterozygote offspring. Okay. Now, in Drosophila, or fruit flies, curled wings is recessive to the normal allele for straight wings. Okay, so let's go ahead and use, well, we usually pick, like, the letters. Like, so if curled is dominant, normally we'd pick a C. But since a large C and a lowercase c look the same, well, then we might use the letter for wings. But then again, a large and W look the same. Then straight also. So all the letters that we would probably use for this are not good choices because the dominant looks like the recessive. So I'm going to use A's. So here, if I say the dominant is the normal allele for straight wings is going to be what's dominant, and curled wings is recessive. Okay, so a homozygous curled, oopsie, I almost did a C. A homozygous curled, oh my goodness, I'm so sorry. A homozygous curled wing male is mated with a homozygous. And then we have here a homozygous straight-winged female. What would the genotype and phenotype be of the F1? So here's our parental generation. Then our F1 cross. Ooh, all of our F1 are heterozygous, and they have straight wings. Now, if we were to say, the F1 are crossed, what happens there, so I'm just going to erase this Punnett square, so we can see that all of our F1 are heterozygous. Now let's take this one step further. If we say a male and female from the F1 are crossed, that means this, because if they just very vaguely say a male and a female are crossed, we know that all of the F1 are heterozygous, so this would be a heterozygous male crossed with a heterozygous female. Now your F2 is where you would have your 3-to-1 ratio for phenotype and then a 1 to 2-to-1 ratio for genotype of your F2 offspring. All right, hairy tongues in the alien race of, I don't know, is a recessive trait to the normal hairless tongues. So this is hairless. And then this is hairy. Okay, oops. If a male who is heterozygous, normal-tongued, has children with a normal-tongued, if a male who is heterozygous, normal-tongued, has children with a homozygous normal tongue, so she's homozygous, so that would be homozygous dominant. What would be the genotypes and phenotypes of their offspring? What percentage would be hairy tongued? What percentage would be normal tongued? So when we look at this, we would see that all of their kids are going to actually have hairless tongues, because the hairless is dominant. And if we look at this one parent who has the normal tongue, it has a recessive, it has all of the offspring are going to inherit this parent's alleles, because if we look at Mendel's law of segregation, when this diploid parent makes gametes, half of the gametes will have a dominant H, and so will the other half. So if this was like a female, every single egg she ever makes will have a dominant H. So every single child she ever has will have a dominant H. Okay, so now dihybrid crosses. In dihybrid crosses, we're looking at 2 traits. So this is asking what would be the expected phenotypic ratio of the F1 offspring if a heterozygous, tall, round pea plant pollinated itself. So it says that tall is dominant to short, and it says that round is dominant to wrinkled. Okay, so here, when it says pollinated itself, that means it's crossed with one like itself, the exact same genotype. So when we go to set up our dihybrid cross, this is where we have our 16 boxes. Okay, so to make our first set of gametes, here this dominant allele is possible to end up with a capital R, but it could also end up with a lowercase r. Then it can the little t can end up with a dominant R, and a little t can end up with a little r. These are all the different possibilities in gametes if this was or due to independent assortment. Now, cool thing, the same is going to hold true for the other side. A dominant T can end up with a dominant R, and then a dominant T can end up with a recessive R, all depending on how the chromosomes, the chromosomal pairs, the homologous pairs, move to the middle of the cell when it goes to meiosis to make these gametes. Okay, then we go ahead and fill in this Punnett square. It's like saying if this was a sperm and this was an egg, if they got together, you'd have 2 large T's and 2 large R's. I'm going to hit pause in my video and then fill this in so I don't take up your time. And then when the video starts playing again, it'll be solved. Now this is asking for the phenotypic ratio of the F1 offspring. So when we find the phenotypic ratio, what we're looking for, first we're going to look at all of the individuals that are dominant for both traits. So that would make tall and round offspring. So we find 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9. And then we want to look for individuals that have tall for one, like dominant for one, but then recessive for the second trait. So that's 1, 2, and 3. And then we're going to look for individuals that have maybe recessive for the first trait, short, but then dominant for the second trait. So that would be 1, 2, and 3. And then we're going to look for, lastly, the individuals that are homozygous recessive for both. So they would be short and wrinkled. And that is 1. So our phenotypic ratio is a 9 to 3 to 3 to 1. Okay. All right. Long legs in racing horses is dominant to short legs. So we're going to write that down. Long legs. And then we have short. And then good muscle tone, so we're going to call that strong, is dominant to weak. Okay. Determine the phenotypes of the F1 generation from a resulting cross between a heterozygous, long-legged, weak-muscled horse. Okay, well, so to get a weak horse, we already know that's 2 lowercase g's, and then this tells us that it's heterozygous for long legs. So here's one parent. And then the next one says a heterozygous, long-legged, strong horse. Now you might be thinking, well, is that strong horse homozygous dominant, or is it heterozygous? Well, because it says heterozygous right here, we're going to assume it's heterozygous for both long legs and strong horses. So we're going to go like, we're going to go like this. And so determine, so what's the F1? So when we set up our Punnett square, we're going to go here. I'll use a different color. So this dominant L can go, oh, that didn't work. Dominant L can go with, okay. Or it can go with that one. And then lowercase. Ooh, like that. Now on the side, we can go here. You can also go with that one. And then this. And that. And then you would fill them in. Okay? So when you fill them in, pretend like each one is a gamete getting fertilized. Okay, and then you continue on. So it's asking for the phenotypes. So these different choices that you get, you would look through them and you would find your outcome. So, yeah, and then determine the phenotypes of the offspring if the genotypes of the mare was this crossed with this. Well, and for this Punnett square, if we were, like, right here, this second horse would be identical to what we wrote right here. So to set up this one, the gametes across the top would look like this. Okay. And our last one, in poultry, a crested head is produced by a dominant gene C, and a plain head is its recessive allele. So crested head is dominant, and then a plain head is recessive. Black feathers is dominant to red feathers. Okay. And a homozygous black-feathered plain-headed bird. Okay. So homozygous for black feathers, and it's plain-headed, is mated with a homozygous red-feathered crested bird. So now the crested could either be dominant or heterozygous, but because it says homozygous here, we're going to assume it's also like that. Determine the F1 genotypes and phenotypes. Well, I already know by looking at this that if I were to take just the B's, all of the offspring would look like this, heterozygous. And then if I took the C's, all of the offspring would be heterozygous. So there's our parental, there's our F1. And now for B, if the F1 birds are allowed to mate with each other, what phenotypic and genotypic ratios are expected from this generation? Well, we've kind of already learned when you cross two parents that are heterozygous for both traits, you're going to get a 9 to 3 to 3